A gene is a fundamental biological unit of heredity. The entire genome of an organism is made up of a number of genes, and a gene usually codes for a single character. The gene is now defined as a sequence of DNA which code for a molecule which is transported to another part other than the site of its synthesis for its biological action. The gene as a whole is a complex structure, but prokaryotic genes are simpler than eukaryotic genes. The eukaryotic genes are monocystronic and prokaryotes are polycystronic. Now, what is monocystronic and polycystronic gene? Monocystronic genes are the genes which code for a single protein, and polycystronic genes code for more than one protein. Different proteins coded by polycystronic genes are often functionally related. A gene is made of two important regions, the regulatory region and the transcriptional unit. The regulatory region contains the sequences essential for the transcription of the gene but are not transcribed themselves, while the transcriptional unit is transcribed to form a RNA molecule. The transcriptional unit starts from a transcription start site. This is the position where RNA polymerase initiates RNA synthesis during transcription, and this nucleotide is designated as the first nucleotide. Among the number of different elements present in this region, the promoter is the most important regulatory element. RNA polymerase binds with gene by the help of promoter and guides it to start transcription. Now, what is a promoter? Promoter is defined as a sequence of DNA having the signal which directs the proper binding of RNA polymerase to DNA and activates it to a form which is capable of initiating the transcription. Some genes have strong promoters that bind the transcription machinery well, and others have weak promoters that bind poorly. Low rates of transcription have been found with weak promoters as compared to the strong promoters. This is because the transcription machinery binds to them and initiates transcription less frequently. Other possible regulatory regions include enhancers, which can compensate for a weak promoter. Eukaryotic promoter regions are much more complex and difficult to identify than prokaryotic promoters. The promoters have two regions of highly conserved sequence with a recognition site and RNA polymerase binding site. A recognition site, which is far from the transcriptional unit, is recognized by the RNA polymerase and facilitates its binding to the template. The RNA polymerase binding site is closer to transcription initiation site and helps in the initiation of RNA synthesis by opening of the double-stranded DNA. Now, prokaryotic promoter. In prokaryotes, the first region, which is highly conserved in most of the promoters, has a consensus sequence T, T, G, A, C, A. There are two regions of -35 region and the -10, which are important in prokaryotic promoter region. The -35 region is normally present between 30 to 40 nucleotide upstream of the transcription initiation site. This sequence is centered about 35 base pair before the start of a bacterial gene. It functions in the initial recognition of a gene by RNA polymerase. The first 3 nucleotides are highly conserved and are present in majority of the promoters as compared to the last 3 nucleotides, which are less conserved. The second region is also a conserved hexanucleotide with the consensus sequence TATAAT. It is also known as -10 region or the Pribno box. The Pribno box is absolutely essential to start transcription in prokaryotes. First 2 bases and the last base are present in 90% of the promoters so far studied. The position of this sequence in relation to transcription initiation site is from -5 to -14. The -10 region is highly adenine thymine rich. The melting of adenine thymine base pair, which is having only 2 hydrogen, requires less energy as compared to guanine cytosine, which is having 3 hydrogen bonds. Thus, adenine thymine region easily gets opened and is involved in the formation of open promoter complex during the initiation of transcription. The distance between the -10 and the -35 regions varies from promoter to promoter. In majority of the promoters, it is 16 to 19 nucleotides. Both -10 and the -35 regions are present in majority of promoters, while few promoters may lack either -10 or -35 sequences. Each promoter has got certain degree of variation from the consensus sequence, and the promoter of highly expressed genes should not deviate much from the consensus sequence. For example, the promoter for the tryptophan synthesis and genes coding for beta galactosidase and other enzymes involved in lactose metabolism are some of the strong bacterial promoters and do not deviate much from the consensus sequence. The strength of the respective promoters depends on number 1, deviation of the 2 regions from the consensus sequence, and number 2, distance between the 2 regions. A tag promoter, which is synthetic promoter, has got -10 region of tryptophan promoter and the -35 region of the lac promoter, with a spacer region of 17 bases between the 2 regions. The tag promoter is the most potent bacterial promoter. The regulatory region of an eukaryotic gene is relatively more complex than prokaryotic promoter. 2 basic regions in eukaryotic and prokaryotic promoters are similar in structure but differ in their relative position. The first region, known as recognition region, is similar to the -35 region of prokaryotic promoter and is known as CAAT box. Its consensus sequence is G G C T C A A T C T and is present between the position of -70 to -80. The second region of eukaryotic promoter is septanucleotide and is similar to -10 region of prokaryotes. This region has a consensus sequence of TATA ATAAT and is called the TATA box or Hogness box. This TATA box is located between -19 and -27 nucleotides. This region is also rich in adenine thymine like Pribno box of prokaryotes and helps in melting of double strand DNA during initiation of transcription. The TA box is often surrounded by a guanine cytosine rich region. We have seen that the position of both prokaryotic and eukaryotic promoters is relatively conserved and is located upstream of the transcription unit. However, in some genes, the promoter may be present downstream of the transcription start site and is within the transcription unit itself and is called downstream promoters or downstream promoter elements, DPE. The promoter of Xenopus laevis 5S RNA gene is a good example of downstream promoter. It is located between plus 55 and plus 80 regions. The promoter itself is transcribed and is represented within the RNA. Other downstream promoters are present in VA gene of adenovirus, which is between plus 9 to plus 72. Normally, the RNA polymerase binds to promoter covering about 60 nucleotides and is within the minus 50 to plus 30 region and initiates the RNA synthesis. Now, the question arises how the downstream promoter will allow this to happen. It is possible that the RNA polymerase binds to promoter, then moves back to transcription start site and starts RNA synthesis. Alternatively, the DNA folds back in such a manner that it can start the transcription while bound at the promoter region. The DPE is a distinct 7 nucleotide element that is located at about plus 30, typically from plus 28 to plus 34 relative to the transcription start site. It is present in many but not all promoters and is bound by transcription factor 2D but not by TBP, Tata box binding polypeptide. In addition, nearly all the promoters that have been found to contain DPE-like sequences are TA-TA-less promoter. Mutational analysis of the initiator and DPE motifs revealed that the DPE acts in conjunction with the initiator to provide a binding site for transcription factor 2D in the absence of a TA-TA box. Interestingly, the loss of transcription activity upon disruption of the TA-TA box in a TA-TA containing promoter could be recovered by the insertion of a DPE at a downstream position in the defective promoter. Thus, the DPE appears to be a functionally important conserved downstream core promoter element. In addition to promoter, other regulatory elements such as the activators, silencers, and enhancers are found in eukaryotic gene. The function of these elements is to regulate the level of gene expression. The enhancers are the sequences that raise the gene expression level to many folds. Thus, enhancers are position and orientation independent and have been seen to be present at a distance of thousands of nucleotides from the site of their action. The enhancer of a particular gene, if cloned along with another gene, may stimulate the expression of the receiving gene. Neither the highly conserved sequences nor the precise mode of their action of enhancers is yet clear to biologists. If the position of enhancer is changed experimentally or by making insertion or deletion in the non-coding region of the gene. The enhancer still maintains its activity because it is position and orientation independent. These elements have proved to be very beneficial in achieving high-level expression of foreign genes. It is that part of a gene which starts from the transcription initiation site and ends at the transcriptional terminator. The entire transcription unit is copied to form RNA. The first element in this region is the transcription initiation site, which does not have any long conserved region. It is located between 6 to 9 nucleotides downstream of the Pribno box in prokaryotes. Following the transcription start site, there is a region of varying length and is known as spacer region. In prokaryotes, it is followed by a region which has complementarity with the 3' end of the 16S-rRNA. This region base pairs with 16S-rRNA and thus facilitates the binding of small subunit of the ribosome to the mRNA during the initiation of translation. This region is known as ribosome binding site, RBS, or Shine-Dalgarno sequence, SD sequence. The length of the SD sequences are usually 4 to 9 bases and is located at about 5 to 11 bases upstream of the insertion codon. These sequences help in positioning the 30S ribosomal subunit to the mRNA. Eukaryotic genes do not have any such region. After about 7 to 13 base spacer regions, the insertion codon ATG is present. The distance between SD sequence and ATG plays an important role in the rate of translation. The insertion codon is followed by a coding region of the gene. The coding region is continuous structure in prokaryotes, and a single open reading frame is present which ends in a translational stop codon. In prokaryotes, all the genes involved in the metabolism of related compounds are arranged in a single line to cause sufficient protein synthesis. For example, the enzymes involved in the metabolism of lactose adjacent to one another between the promoter and terminator. By this type of arrangement of genes, one single binding event by the enzyme RNA polymerase is sufficient to cause the synthesis of all relevant proteins, that is, the genes are coordinatively regulated. Such an organized set of genes is known as operon. Eukaryotic genes are much more complex as compared to prokaryotic genes. Accompanying the exons, a large number of non-coding sequences are present in coding region of the eukaryotic gene. These non-coding segments are known as introns and do not code for any polypeptide. Exons are sequences in the gene that are found in the functional mRNA. Exons include coding sequences but may also include non-coding sequences. The beginning of the first exon defines the site of initiation of transcription, since there is no processing of the 5' end of the primary transcript. The end of the final exon defines the site of cleavage of the primary transcript at what is known as the polyadenylation site, creating the mature mRNA 3' terminus. Now, what are introns? Majority of the eukaryotic genes have introns which are present within the coding region of the gene, but are not the part of the open reading frame, ORF. In prokaryotes, They are only found in tRNA and rRNA. These sequences are transcribed and are present in the primary transcript but are not required for the biological function of the mRNA and are excised by a process known as splicing. The number and length of the intron varies from species to species and are alternate with exons. The introns can form up to 75% or more of a gene. For example, the mouse dihydrofolate reductase DHFR gene is 8.1 kb while the mRNA is only 1.6 kb and the coding region is only 558 nucleotides. A number of genes in yeast are continuous and do not have introns. In majority of the mammalian genes, the introns occupy more space than the exons. The presence of a large number of introns help in protecting the genome against spontaneous mutations. It is now recognized that introns are a complex mix of different DNA, much of which are vital to the life of the cell. As their functions are being determined, the relationship of introns to cancer and their use as tumor makers is also being explored. Several functions for introns have already been identified, and evidence for a role for them is indicated by the finding that some introns alteration are directly related to the development of cancer. Early insight in intron function was a result of the finding that many non-coding DNA sequences are not random base pairs but have certain features in common with human language. The finding that introns manifest the same complex pattern of communication found in human speech supports the supposition that they carry functional information. Of course, the existence of a pattern does not in itself provide direct evidence for a function, but it does indicate a potential systematic cause exists which produces the pattern found. Transcription of introns to small regulatory RNA, which is known as microRNA, has been found the most important role of introns.